Hi guys, recent study showed that fasting boosts stem cell regenerative capacity. Age-related declines in stem cell function can be reversed by 24-hour fast. According to a new study, biologists found fasting dramatically improves stem cells' ability to regenerate in both aged and young mice. As people age, their intestinal stem cells begin to lose their ability to regenerate. These stem cells are the source for all new intestinal cells, so this decline can make it more difficult to recover from gastrointestinal infections or other conditions that affect the intestine. This age-related loss of stem cell function can be reversed by a 24-hour fast. According to a new study from MIT biologists, the researchers found that fasting dramatically improves stem cells' ability to regenerate in both aged and young mice. In fasting mice, cells begin breaking down fatty acids instead of glucose, a change that stimulates the stem cell to become more regenerative. The researchers found that they could also boost regeneration with a molecule that activates the same metabolic switch. Such an intervention could potentially help other people recovering from GI infection or cancer patients undergoing chemotherapy. The researchers say fasting has many effects in the intestine, which include boosting regeneration as well as potential uses in any type of ailment that impinges on the intestine, such as infections or cancers, says Umar Yilmaz, an MIT assistant professor of biology, a member of the Koch Institute for Integrative Cancer Research, and one of the senior authors of the study. Understanding how fasting improves overall health, including the role of adult stem cells in intestinal regeneration, in repair, and in aging, is in fundamental interest of my laboratory. David Sabatini, an MIT professor of biology and member of the Whitehead Institute of Biomedical Research, is also a senior author of the paper, which appears in the May 3 issue of the Stem Cell. This study provides evidence that fasting induces a metabolic switch in the intestinal stem cells from utilizing carbohydrate to burning fat, as Sabatini says. Interestingly, switching these cells to fatty acid oxidation enhances their function significantly. Pharmacological target of this pathway may induce a therapeutic opportunity to improve tissue homeostasis in age-related pathologies. The paper's lead author are Whitehead Institute postdoc Maria Mahilova and Koch Institute postdoc Chia-Wei Chang. Boosting regeneration. For many decades, scientists have known that low caloric intake is linked with enhanced longevity in humans and other organisms. Yilmaz and his colleagues were interested in exploring how fasting exerts its effect at the molecular level, specifically in the intestine. Intestinal stem cells are responsible for maintaining the lining of the intestine, which typically renews itself every 5 days. When an injury or infection occurs, stem cells are key to repairing the damage. As people age, the regenerative abilities of these intestinal stem cells decline, so it takes longer for the intestine to recover. Intestinal stem cells are the workhorses of the intestine that give rise to most stem cells and to all of the various differentiated cell types of the intestine, notably during aging. Intestinal stem cell function declines during aging, which impairs the ability of the intestine to repair itself after damage, says Yilmaz. In this line of investigation, we focus on understanding how a 24-hour fast enhances the function of young and old intestinal stem cells, says Yilmaz. Also, after mice fasted for 24 hours, the researchers removed intestinal stem cells and grew them in a culture dish, allowing them to determine whether the cell can give rise to organoids. The researcher found that stem cells from fasting mice doubled their regenerative capacity. It was obvious that fasting had this really immense effect on the ability of intestinal crypts to form more organoids, which is stem cell driven. Mihailova says this was something that we saw in both the young mice and the aged mice, and we really wanted to understand the molecular mechanism driving this. Metabolic switch, further studies including sequencing the messenger RNA of stem cells from the mice that fasted revealed that fasting induces cells to switch from their usual metabolism, which burns carbohydrates, such as sugar, to metabolizing fatty acid. This switch occurs through the activation of transcription factors called PPARs, which turn on many genes that are involved in metabolizing fatty acids. The researchers found that if they turned off this pathway, fasting could no longer boost regeneration. They now plan to study how this metabolic switch provokes stem cells to enhance their regenerative abilities. They also found that they could produce the beneficial effects of fasting by treating mice with a molecule that mimics the effects of PPARs. This was also very surprising. Chang says just activating the one metabolic pathway is sufficient to reverse certain age phenotypes. The findings suggest that drug treatment could stimulate regeneration without requiring patients to fast, which is difficult for most people. One group that could benefit from such treatment is cancer patients who are receiving chemotherapy, which often harms intestinal cells. It could also benefit older people who experience intestinal infection or other gastrointestinal disorders that can damage the lining of the intestine. The researchers plan to explore the potential effectiveness of such treatment, and they also hope to study whether fasting affects regenerative abilities of stem cells in other types of tissues. Thanks for watching.